A shaft of sunlight fractures as it penetrates the reef surface, illuminating a tapestry of coral ridges that sway with the rhythm of a gentle current. Here, in the labyrinthine crevices of staghorn and brain coral, a small assembly of Amphiprion percula, the orange and white banded clownfish, governs its territory with the precision of seasoned diplomats. Their world is defined by the undulating tentacles of the sea anemone, whose stinging cells both endanger and protect. As you glide closer, suspended in the cerulean hush, you can almost hear the faint hum of microscopic life teeming around every polyp, and the soft snap of crustaceans foraging in the shadows. This is not a scene of simple survival, but of an intricate negotiation between two vastly different organisms, one driven by the ancient chemistry of trust, the other by a hunger that can only be sated by others' demise. A lone clownfish darts through the anemone's embrace, its skin slick with a secret defense, a specialized mucus that cloaks it from the anemone's nematocysts. Rather than triggering a lethal cascade of stings, this mucus acts as a biological olive branch, signaling friend, not food, to the anemone's quivering arms. Scientists studying this phenomenon have found that the mucus lacks the specific compounds, peptides and lipids, that normally activate the anemone's discharge mechanism. In effect, the clownfish has learned the language of its host, rewriting the chemical script that would otherwise end in venomous demise. Watching this beneath the waves, you sense a silent mutual recognition. Neither creature truly owns this domain alone. Together, they form a coalition far stronger than any solo sentinel. Within this alliance, nutrients flow both ways in a delicate equilibrium. As the clownfish patrols its anemone enclave, it chases away predators, sometimes tiny wrasse or damselfish foolish enough to snack on tentacles, incurring the host's protection in return. Each passing fin leaves behind a trace of ammonia-rich waste, which in turn becomes sustenance for the anemone's symbiotic algae. These algae, nestled within the anemone's tissues, carry out photosynthesis under the reef's shimmering canopy, converting sunlight into vital sugars. Without the clownfish's contributions, the anemone's primary food source would wane. Without the anemone's shelter, the clownfish would be exposed to every prowling grouper or moray eel in the neighborhood. It is an exchange rooted in survival, where chemical whispers translate into ecological stability, and yet it balances on a knife edge. Too many clownfish can overwhelm an anemone, stripping its tentacles bare, while too few leave it vulnerable to algal overgrowth and bleaching. Beneath this balanced surface, the politics of clownfish society unfold with startling complexity. A clutch of eggs, pinkish and oval, adheres to a flat rock at the periphery of the anemone, guarded vigilantly by the dominant female, whose larger size and vibrant stripes signal her reign. Nearby, the male performs his nightly ritual, fanning the eggs with broad, rhythmic strokes of his pectoral fins to ensure oxygenation. Among the juveniles lurking in the tentacles, a strict pecking order prevails. The largest non-breeder holds second rank, while the smallest remains at the bottom, its growth stunted by continual harassment. This hierarchy is no mere display of braggadocio, it dictates survival. When the female perishes, whether in a surprise predator encounter or through disease, the largest male undergoes a remarkable transformation, shedding testicular tissue and cultivating ovaries in a process triggered by the absence of female pheromones. Within weeks, he assumes her mantle, complete with the physical attributes necessary for egg production. The cycle persists, ensuring that the anemone's chamber always hosts one fertile female, one protector male, and a cadre of hopeful juveniles awaiting their turn. The reef does not tolerate stagnation. Seasonal temperature spikes, subtle yet relentless, sometimes push the anemone toward bleaching, expelling its vital algae and paling to ghost white. In such moments, the clownfish engages in an unexpected behavior. It ferries fragments of healthy algae from nearby corals, brushing them against the anemone's base in futile attempts at recolonization. Though these efforts may seem quixotic, researchers have observed that anemones occasionally accept these unsolicited grafts, restoring their photosynthetic partners and staving off collapse. It is a testament to the clownfish's role not merely as a tenant, but as an active steward, one whose behavioral repertoire extends beyond defense into ecological maintenance. Predator-prey dynamics introduce further twists to this tableau. While the anemone's stinging arms deter most predators, some cunning filefish and butterflyfish have evolved resistance to its nematocysts, nibbling on tentacles with surprising agility. In these encounters, the clownfish slots into the role of frontline guardian, darting out in flashes of orange and white to drive off intruders far larger than itself. Yet even here, irony lingers. A hungry anemone might snap at the dirty, oily sheen of the clownfish's mucus if its protective filter breaks down, reminding us that mutualism requires constant upkeep. Too many assaults by invaders can diminish the anemone's health, prompting shifts in the clownfish's behavior, from frenetic defense to cautious reconnaissance as the pair negotiate the shifting terms of their treaty. This microcosm, less than a square meter of reef, holds broader lessons for the entire coral community. Wherever clownfish-anemone partnerships thrive, fish biodiversity increases, and the structural complexity of the reef strengthens. Sheltered nooks provide refuges for snails, shrimps, and juvenile wrasses, while the chemical byproducts of this partnership circulate through the water, influencing algal growth and oxygen levels. In bleaching events, areas dominated by healthy anemone-clownfish alliances recover more swiftly, aided by the mutualists' localized nutrient recycling. Conservationists now recognize that protecting these small ecosystems may offer outsized benefits, acting as seed banks for broader reef restoration. As the current shifts once more, the clownfish family retreats deeper into the anemone's embrace, its top-ranked female pressing gently into the crenellated swirl of tentacles. Even here, motion persists. Eggs pulsate in rhythmic synchrony with the female's breathing, while the male keeps watch at the edge, eyes flicking with a predator's caution. Above them, particulate matter drifts like stardust in a blue void, and somewhere beyond the visible reef, a reef shark glides by in ghostly silence, unaware of the fragile accord thriving below. This is the world of the clownfish, an ongoing pact forged in mucus and calcium, light and shadow, where trust emerges from ancient biochemical dialogues, and the very future of the reef may hinge on the actions of these diminutive sentinels. In their tale, one finds not just the story of a fish and its host, but a narrative of resilience, adaptation, and the subtle bargains that sustain life beneath the waves. A dappled shaft of sunlight penetrates the reef's canopy, scattering across a mosaic of finger coral and encrusted limestone as a solitary blue tang glides into view. Its body, a vivid sapphire streaked by a bold black curve, moves with precision through the labyrinthine grooves of the reef crest. Each flick of its tail reveals a flash of brilliant yellow, a warning flare against the cerulean palette. In this realm of constant motion, the blue tang is both surgeon and steward, wielding a retractable spine at the base of its tail like a hidden scalpel, ready to trim and carve the living canvas of the ocean floor. Invisible to most eyes, the tang's scalpel is a marvel of evolutionary engineering. Embedded within a flexible sheath of skin, two razor-sharp spines lie folded against the caudal peduncle. When threatened or asserting dominance, the fish engages a specialized ligament mechanism that swings the spines outward, locking them into a defensive posture. Observers using high-speed videography have recorded the spines deploying in mere milliseconds, faster than the blink reflex of a human. Yet this weapon is more than a deterrent. It is a tool. As the blue tang maneuvers close to a patch of turf algae, it braces itself against a coral branch and deploys the spine to steady its approach, anchoring precisely as its small brush-like teeth shave filamentous algae from the substrate. This grazing behavior is the reef's equivalent of urban landscaping, preventing algal blooms that can smother coral polyps and disrupt the broader ecosystem. Overgrown algae pockets, left unchecked, cast shadows that hinder the photosynthetic machinery within coral tissues, leading to weakened structures and increased susceptibility to disease. The blue tang's feeding scars often resemble the swooping arcs of a gardener's shears, each pass exposing the vibrant pink and white skeleton of coral that had been cloaked in green growth. Through countless generations, reef health has become intertwined with the tang's persistent trimming, an uncelebrated act of maintenance that underpins much of the reef's biodiversity. As the tang pauses mid-graze, a faint scent of citrus and must drifts in the current, marking its chemical defenses. Beneath its scales lies a suite of bioactive compounds, bile-derived terpenoids and cardiac glycoside analogues that render its flesh unpalatable to most predators. When a hungry grouper or snapper dares to test this armor, it discovers a bitterness so intense that it quickly releases the tang, spasms rippling through its stomach. Researchers capturing this reaction in controlled environments noted that predatory fish will often retch and avoid anyone bearing the blue tang's signature hue thereafter. In this way, the tang's colors serve not only as camouflage among shadows, but as an unmistakable aposematic signal, warning onlookers of a chemical arsenal beneath the surface's beauty. Drifting further along the reef slope, the reef topography transforms. Sponges bloom like underwater flora, and columns of butter-soft corals sway in unison. Here, a small school of blue tangs performs synchronized passes, each individual maintaining a precise distance to avoid accidental spine-lock clashes, while juvenile tangs sport more subdued patterns, pale blue bodies ringed by a darker band. Mature specimens exhibit the full intensity of their colors, a sign that they have assimilated enough algal toxins to potentiate their defenses. In juvenile stages, they remain more cryptic, lingering near branching corals or hiding within the folds of massive coral heads, minimizing exposure to predators while they grow and accumulate their chemical store. Beyond its role in reef maintenance and self-defense, the blue tang has become an unwitting ambassador in the global aquarium trade. Its electric coloration and compact size make it a coveted specimen for hobbyists, and each year, hundreds of thousands are collected from reefs in the Indo-Pacific. The harvesting process, often conducted by divers using cyanide to stun fish, can devastate sections of the reef, killing non-target species and compromising the living architecture that supports myriad creatures. Even with improved net capture techniques and best practice guidelines, mortality rates during transport can exceed 20%, as stress and injury take their toll. Conservationists warn that without stringent quotas and robust enforcement, wild populations of blue tangs could decline sharply within a decade, leading to cascading effects on reef health. Within marine protected areas that prohibit wild collection, anecdotal reports from divers and scientists hint at localized algal blooms where tang densities are lower. Patches of turf algae proliferate unchecked, forming dense mats that smother small corals and alter sediment dynamics. In one study conducted off the coast of Samoa, sectioned off reef plots devoid of blue tangs saw a 35% increase in algal cover within 6 months, a stark contrast to adjacent areas where tangs continued their ceaseless grazing. These observations underscore the tang's integral ecological function and raise pressing questions about the sustainability of current collection practices. Yet the blue tang story also reflects resilience and adaptation. Captive-bred tangs, once a rarity, have become more commonplace in research facilities and aquaculture ventures. Through careful manipulation of diet and environmental variables, breeders have succeeded in coaxing tangs through their full life cycle, from egg to adult, a process that once eluded scientists due to the larvae's sensitivity to water quality and the difficulty of replicating the complex algal diets they require. While still in its infancy, this aquaculture approach promises to relieve pressure on wild stocks and offers scientists an unprecedented opportunity to study tang chemistry, behavior, and genetics without the ethical and ecological costs of wild capture. As afternoon light begins to wane, the reef takes on a cooler hue and shadows lengthen across the swaying corals. In this shifting palette, the blue tang moves into deeper water, its silhouette a precise wedge slicing through indigo gloom. Here, a lone juvenile seeks refuge beneath the flared canopy of a Gorgonian sea fan, its pale flanks and dark band blending seamlessly with the twilight reef. Scientists believe this transitional refuge allows young tangs to acclimate to the lower light conditions of adulthood before they join the open water grazers above. Observing this migration, one senses the tang's life as a continuum of challenges, evading predators, maintaining reef health, and managing its own population dynamics. Predator interactions introduce further surprises. Though most fish retreat at the taste of tang toxins, some triggerfish species appear indifferent, employing crushing jaws to bite through the tang's armor, spines, and all. In such confrontations, the blue tang's only escape is speed and agility, darting into complex reef structures where larger predators cannot pursue. These pockets of refuge, the overhangs, tunnels, and crevices, serve as blue tang nurseries, safe zones where juveniles can learn the art of grazing and spine deployment under the watchful eyes of older tangs. The dynamics of these nurseries and how they shift with changing reef structures remain an active field of study, hinting at deeper layers of social learning within fish communities. Farther along the reef crest, a research station glints beneath passing waves. A cluster of sensors records algae growth rates, water temperature, and fish movement. Remote monitoring has revealed that blue tang grazing activity peaks during mid-morning and late afternoon, times when algal productivity is highest, and diminishes during the warmest part of the day, likely as a behavioral adaptation to avoid thermal stress. These data also show that tangs adjust their grazing routes in response to storm damage, favoring newly cleared surfaces until coral regrowth encroaches once more. In every shift and nuance, the blue tang reveals itself not as a mere fish, but as a dynamic agent whose behaviors ripple across the reef ecosystem. By the time dusk settles over the reef, the blue tang school comes alive in a final bout of maintenance, racing along ridges and walls in a synchronized ballet of feeding and defense. Their bodies momentarily form a moving tapestry of blue and black, punctuated by the gleam of deployed spines, each fish aware of its neighbor's position to avoid accidental clashes. As the last rays of sunlight fade, the school disperses, each fish seeking the shelter of crevices and underhangs, spines retracted, bodies still streaked with the day's chemical arsenal. In this hour of transition, one can reflect on the blue tang's dual identity: gardener and warrior, chemical alchemist and ecological engineer. Its presence shapes the reef's very foundation, trimming algae to free space for coral larvae and deterring predators through both blade and bile. Yet, equally, it stands as a testament to human impact, both as a victim of overcollection and a potential beneficiary of sustainable aquaculture. Within the blue tang's razor-edged tail and luminous body lies a narrative of balance and consequence, reminding us that the reef's future is inseparable from the fate of its smallest, most skilled inhabitants. Beneath a sky rippling with midday light, the reef's spires and plateaus extend like an undersea metropolis, their contours sculpted by countless generations of coral growth. Amid this bustling cityscape, a queen angelfish emerges from a cranny between two massive porites colonies, its body a kaleidoscope of azure, gold, and violet. The fish moves deliberately, its pectoral fins fanning with precise strokes that disturb the suspended plankton and set motes of sand drifting like snow. Every subtle adjustment of its sleek form speaks to a creature attuned to both the reef's architecture and its hidden tensions. Here, the angelfish is both caretaker and opportunist. It hovers close to the coral surface, reducing the spread of fast-growing filamentous algae with targeted nips, yet at times extends its narrow, protrusible mouth to pluck delicate polyps from stony corals. In this paradoxical feeding behavior, the angelfish balances the reef's need for cleanliness against its own nutritional demands, trimming away suffocating algae while occasionally taking a bite from the very foundation it protects. As the angelfish completes a pass along a coral ledge, a rival flash of color appears from the opposite direction, a regal angelfish whose vivid bands seem to glow against the reef's shadowed alcoves. The two converge in a corridor of branching coral, their bodies undulating in unison as they size each other up. Neither strikes immediately. Instead, they engage in an elaborate display of vertical fins, erecting and folding, subtle body tilts that reveal alternating stripes and spots. In these ritualized encounters, color is as much weapon as warning. The sudden flare of yellow dorsal spines signals intent to defend territory. Onlookers, surgeonfish and parrotfish grazing nearby, pause in their own routines, creating a hush that amplifies the crackling sound of displaced water. Only when the dominant individual commits with a quick thrust of its head does the contest begin, both fish lunging forward in mirrored motions. Within seconds, the invader retreats, slipping away into the labyrinth as the victor settles back into its patrol, the reef's rhythm restored. Beyond these duels lies a broader drama that unfolds on ocean currents hundreds of meters above the reefscape. During breeding season, female angelfish ascend in solitary spirals, releasing buoyant egg ribbons into the water column. Each egg, barely visible to the naked eye, contains oil droplets that keep it suspended, a tiny vessel set adrift on the ocean's conveyor belt. Currents carry these eggs across vast distances, sometimes spanning entire archipelagos, before they hatch into translucent larvae. In the dim pelagic zone, these larvae ingest microscopic algae and zooplankton, gradually accumulating the pigments and energy reserves they need for settlement. Weeks later, metamorphosis transforms these drifting forms into miniature versions of their parents, complete with scaled-down fins and the first hints of adult coloration. When they finally descend, they seek out suitable reef habitat, guided by chemical cues and acoustic signals emanating from the communal bustle below. In this way, a single reef's angelfish population remains genetically connected to distant reefs, weaving a living network that spans thousands of kilometers. This life cycle, so dependent on both local reef health and the broader oceanic environment, renders angelfish particularly sensitive to ecological disruption. Their presence, diversity, and behavior provide researchers with invaluable insights into reef conditions. In regions scarred by pollution or bleaching, angles of absence appear. Fewer young recruits explore the rubble fields. Fewer established adults patrol the coral faces. Conversely, where water clarity and nutrient balance remain within natural thresholds, angelfish thrive, their numbers swelling to form dense patrols that keep algae in check and help maintain coral vitality. During a recent survey in the Coral Triangle, scientists noted a strong correlation between angelfish biomass and rates of coral recruitment, suggesting that the fish's dual role as cleaner and selective feeder creates the optimal substrate for new coral polyps to settle. In effect, the angelfish acts as a living barometer, its fortunes rising and falling in concert with the reef's own fortunes. Returning focus to the angelfish in its microhabitat, you observe a subtle shift in behavior as dusk approaches. The fish transitions from its daytime routine of grazing and vigilance to a more subdued posture, gliding into the overhangs cast by table corals. Here, it finds a crevice just large enough to accommodate its flattened body, retracts its fins, and remains motionless. Surrounding it, cardinal fish and shrimp gobies take their turn as reef sentinels, while the angelfish's cryptic coloration shifts from vivid dayglow to muted camouflage under falling light. Even in this quiet state, its presence exerts influence. The fish's shadow deters small invertebrate predators from probing the coral, and its waste continues to fertilize the microscopic algae that feed coral symbionts. This seamless transition from active guardian to passive protector underscores the angelfish's versatility and the many layers of interaction that define its existence. In the end, the angelfish stands as a testament to the reef's complexity, a creature whose beauty belies its essential contributions and paradoxes. Its feeding paradox, oscillating between coral nibbler and algal gardener, reflects the intricate balancing act that sustains the reef. Its territorial confrontations reveal a world governed as much by visual language as by violent struggle. Its life history, flowing from reef to open ocean and back again, maps the unseen highways that unite coral ecosystems across oceans. And its role as bioindicator offers a living gauge of reef health, guiding conservation efforts with every flick of its vibrant fins. Within the angelfish's movements and behaviors lies a lesson in coexistence, that the reef's survival depends on the nuanced interplay of roles, and that even the most striking guardian can be both protector and predator in the same breath. Beneath a shifting canopy of fractured sunlight, the reef floor unfolds as an ever-changing mosaic of color and form, where every crevice and ledge serves as both stage and shelter. Here, amidst the waving plumes of soft corals and the stubby fingers of porretes, a small shoal of butterflyfish drifts in unison. Their bodies, no more than 15 centimeters in length, bear a riot of stripes and spots that slice through the water like the brushstrokes of an impressionist painting. As you follow their movement, the scene resonates with a subtle tension. These pretty fish are anything but passive. In fact, the spots and bands that embellish their scales are weapons of deception so refined that scientists still uncover new layers to their significance. Each stripe breaks up the contours of fin and flank, hiding the butterflyfish from both predators and prey as effectively as a darting wink across a crowded room. This phenomenon, disruptive coloration, becomes evident when a prowling trevally slips into view, its silvery flank reflecting the reef's dappled light. The trevalle's eyes lock onto the shoal, and its body tenses for the strike. Yet as the predator lunges, the butterflyfish respond with a sudden scatter, their stripes fracturing their outlines into a dozen shards of color. The trevalle's swift lunge miscalculates, its jaws snapping shut on empty water, a fraction of a second too late. Under high-speed photographic analysis, researchers have observed that the predator's visual system struggles to track the fast-moving seams of black and white. Each band creates a false boundary, a visual glitch that confuses depth perception. In one experiment, predation attempts on artificially patterned butterflyfish decoys dropped by nearly 40% compared to solid-colored controls. Such precision makes clear that these fish do not merely blend in, they engineer their concealment with mathematical exactness. Within this landscape of optical trickery, another level of deception takes shape. False eye spots. On the narrow posterior near the base of the dorsal fin, A dark circle ringed in bright white mimics the fish's actual eye, a ploy that invites predators to strike at non-vital regions. In the hush before nightfall, a solitary angelfish drifts closer to a feeding butterflyfish. As the angelfish launches its lunge, the butterflyfish pivots, presenting the eye spot to its attacker. The angelfish's strike corners toward the tail, where the hardened spines bristle as a defensive buffer. In the aftermath, the butterflyfish darts off, its true eye concealed behind a vertical stripe that bisects its head, further obscuring its identity. This twofold system, stripe masking and eye spot misdirection, operates as an integrated defense, honed by millennia of predator-prey arms races. Amid these high-stakes encounters, an unexpected alliance unfolds at the reef's famed cleaning stations. Here, porcelain crab-colored shrimp cluster among the waving tentacles of tube sponges, their claws poised to nip away parasites. Butterflyfish queue in orderly fashion, hovering headfirst in precise orientation. The largest individuals drift nearest the shrimp, while smaller juveniles wait their turn farther back. As the shrimp dart between scales, tapping gently to signal readiness, the butterflyfish adopt a rigid posture, their fins held flush to their sides. In this moment, the fish's bold patterns, once tools of concealment, become open invitations, bright markers that guide the cleaners to the sites of infestation. Microscopic copepods and isopods, invisible in the daytime swirl, emerge in the shrimp's pincers, plucked free from gill arches and skin folds. Through this mutualistic exchange, butterflyfish rid themselves of harmful hitchhikers, while cleaner shrimp gain sustenance. Observers note that this partnership can reduce fish parasite loads by up to 90%, bolstering butterflyfish health and longevity. Yet the reef's currents carry both boon and threat. Warming seas have triggered mass coral bleaching events, whitening vast swathes of reef and stripping away the structural complexity that underpins every cleaning station. As corals fade to ghostly skeletons, the tube sponges that harbor cleaner shrimp dwindle, disrupting the symbiosis that supports butterflyfish survival. In bleached zones, butterflyfish patrol longer distances, their usual appointment-based cleaning ritual replaced by frantic scrub and flea scavenging as they seek parasite relief. Scientists monitoring these shifts report a 25% increase in parasite-related morbidity among butterflyfish populations in degraded reefs, a stark contrast to their behavior in healthy habitats where cleaning occurs daily. The loss of patterned refugees further exacerbates their vulnerability. Without the vibrant tapestry of living coral, the disruptive stripes and eye spots lose contrast, rendering the fish more conspicuous against the now monochrome backdrop. Even as the reef's visual environment unravels, butterflyfish reveal surprising behavioral adaptations. In some degraded areas, individuals have been observed congregating in small groves of macroalgae, using the fronds as makeshift camouflage. Their stripes, which once mimicked the sway and shadow of coral branches, now align with strands of halimeda and sargassum, creating a novel blend of body pattern and plant form. This rapid behavioral shift highlights the fish's capacity for ecological flexibility, yet it also underscores the fragility of their equilibrium. Macroalgae patches are temporary and unstable, prone to washout during storms, and they offer limited feeding opportunities compared to the cleaner stations and diverse prey of healthy reefs. As a result, butterflyfish engaging in this improvised concealment often exhibit stunted growth and reduced reproductive success, a trade-off between immediate survival and long-term fitness. Predator-prey dynamics continue to evolve in unexpected ways. A recent study documented a coral hawkfish that had learned to exploit false eye spots by preferentially targeting the rear of butterflyfish, using the non-vital bite to injure rather than kill. The hawkfish's jagged bite scars appear as pale crescent marks near the eye spot, evidence of an emerging behavioral arms race. During day-long observations under lowered humidity diving conditions, researchers recorded that butterflyfish bitten in this manner displayed a compensatory behavior. They swiftly adjusted their spacing within the shoal, retreating from open areas to denser coral thickets where hawkfish access is limited. This self-organizing group movement illustrates a collective defense mechanism, one driven not by instinct alone, but by rapid social learning and spatial awareness. Meanwhile, climate-driven ocean acidification affects shell-forming organisms, including the crustaceans that butterflyfish prey upon. When copepod shells thin, their bodies become more brittle and less nutritious, leading butterflyfish to alter feeding strategies. Instead of standard pecking and suction techniques, some species employ coordinated crowd tactics, where multiple fish converge on a patch of substrate, creating vortices that dislodge invertebrates en masse. These tactics shift the functional role of butterflyfish from selective feeders to ecosystem engineers of a different sort, reshaping sediment and microhabitat structure. Although such behavior underscores the fish's adaptability, it also signals the cascading effects of altered prey physiology on higher trophic levels. Returning focus to the reef's living drama, the sun dips toward the horizon, casting long, wavering shadows across the coral slopes. Butterflyfish glide into twilight, their vivid patterns dimming into muted arrays of grey and black as their chromatophores respond to fading light. Under the cover of dusk, they form tighter shoals and seek out the last cleaning shrimp still emerging from their sponges. The reef's chorus dims to a soft susurration of nocturnal invertebrates stirring among the rubble. In this final hour, the full spectrum of butterflyfish survival strategies, camouflage, mimicry, cooperation, and innovation, unfolds in a seamless performance that ties together the reef's many interdependencies. In every stripe and spot lies a story of deception, adaptation, and cooperation, revealing how a seemingly delicate fish wields pattern as both armor and invitation. From thwarted trevally strikes to the precise choreography of cleaning stations, butterflyfish demonstrate that survival in the coral realm requires both art and science, instinct and ingenuity. Yet their fate remains bound to the reef's health. As the tapestry of living coral recedes, the delicate balance of patterns and partnerships unravels, leaving these pretty fish exposed in ways no disruptive coloration can entirely conceal. Their perseverance offers a testament to nature's inventiveness and a warning that the loss of their environments may strip away the very stage on which they perform their extraordinary acts of deception. Beneath the wavering light of dawn, the reef's contours emerge from the deep in sharp relief, as if sculpted by unseen hands during the night. Here, among towering plate corals and the tangles of branching staghorn, a school of parrotfish moves with deliberate purpose, their vibrant hues of green, blue, and pink streaking past like living paint smeared upon the rock. Each fish advances headfirst toward a protruding coral spur, lowering its beak-like dental plates to the surface. With a grinding crack that resonates through the water, the parrotfish shatters the coral's limestone structure, scraping away fragments of polyps and calcium in a single motion. Observing this from a respectful distance, one realizes that these bursts of destruction are not mere feeding frenzies, but intricate acts of bioerosion that reshape the reef's very architecture. The parrotfish's beak is a marvel of natural engineering, a fused set of teeth honed over millennia to deliver maximum crushing force in rapid, repeated bites. Unlike the sharp, piercing dentition of predators, these plates interlock to form a durable mortar capable of pulverizing hard coral into digestible grains. Internal pharyngeal teeth then further reduce the fragments, breaking them down into fine particles that pass through the digestive tract. In this process, each individual parrotfish processes hundreds of coral bites per hour, and over the course of a year, a single large specimen can produce upwards of 2 tons of sand. This sediment, expelled in clouds of white grains, drifts on the current before settling into the reef's crevices and nearby sandy lagoons. In effect, parrotfish serve as both demolition crew and sand makers, converting living reef into the building blocks of beaches and back reef habitats. As sunlight climbs, illuminating the reef's hidden valleys, the significance of this bioerosion becomes clear. Parrotfish modulate the reef's topography, carving channels and smoothing abrupt edges, which in turn influence water flow, sediment deposition, and coral larval settlement. Areas heavily grazed by parrotfish show gentler slopes and fewer deep crevices clogged by dead coral rubble, creating hospitable zones for juvenile corals and sponges. In contrast, regions where parrotfish have been overfished reveal blocky, uneven landscapes where sediment accumulates in mounds, smothering potential settlement sites and reducing overall biodiversity. Researchers measuring carbonate budgets on Caribbean reefs have documented declines of up to 70% in reef growth rates when parrotfish populations dwindle, underscoring their essential role as ecosystem engineers. By midday, the reef's community buzzes with life. Damselfish guard their turf gardens, wrasses dart in pursuit of parasites, and moray eels peer from cavernous holes. Yet as evening approaches, the parrotfish undertake another extraordinary feat. One by one, they begin to secrete a translucent mucus cocoon around their bodies, enveloping themselves in a shimmering sphere nearly as large as the fish itself. Within this gelatinous shell, the parrotfish rests, insulated from parasites and predators alike. The cocoon acts as an antioxidant barrier, neutralizing harmful free radicals produced during the fish's intense daytime grinding, and it masks chemical cues that nocturnal hunters, such as snappers and sharks, use to locate prey. Under low light, these floating capsules drift among the coral pinnacles, resembling ethereal balloons, their occupants lulled into a state of near immobility until the first hints of dawn dissolve their protective veil. This nightly ritual, captured by time-lapse underwater photography, reveals an unexpected vulnerability. Sudden temperature anomalies can disrupt cocoon formation, leading to increased parasite infestations and predation. In regions where water quality suffers from runoff or pollution, parrotfish have been observed secreting thinner, less cohesive cocoons, leaving them exposed to attack by eel-like wolf eels or barracuda. These localized pressures further compound the loss of parrotfish in degraded reefs, initiating feedback loops where fewer grazers lead to coral overgrowth by macroalgae, which in turn lowers water quality and impairs cocoon production. The result is a cascade of ecological breakdown, where the vital link between parrotfish behavior and reef resilience unravels. The human dimension of this paradox comes into stark relief when one considers the fate of tropical coastlines that rely on parrotfish-generated sand. On islands where fishing is unregulated, parrotfish are often targeted for their size and striking colors, commanded as prized catches in local markets. As populations shrink, beach erosion accelerates. Sandbanks that once buffered storm surges recede, and shoreline communities face heightened vulnerability to hurricanes. In the Maldives, studies have shown that beaches backed by reefs with intact parrotfish populations erode at half the rate of those in parrotfish-depleted zones. Similar patterns in the Caribbean have prompted governments to impose fishing bans on key parrotfish species, recognizing that the survival of coastal infrastructure and tourism hinges on maintaining these natural sand factories. Yet even well-intentioned conservation measures encounter cultural complexities. In some Pacific Island communities, parrotfish hold deep traditional value, their harvest tied to social rituals and ceremonies. Conservationists working alongside local fishers have found that collaborative approaches, such as seasonal no-take zones and educational campaigns highlighting parrotfish's role in beach formation, yield more sustainable outcomes than top-down regulations. In Fiji, community-led management plans that incorporate parrotfish spawning aggregations have led to notable recovery of both fish biomass and shoreline stability, demonstrating that socio-ecological integration is essential in preserving the reef's engineers. Returning to the reef's present state, one can appreciate the parrotfish as a linchpin species whose every bite carries the dual potential for destruction and creation. Its beak, though capable of brutal pulverization, lays the groundwork for new coral recruits. Its sediment production builds beaches far beyond the reef's embrace. Its nightly cocoons safeguard its own survival and, by extension, the reef's continued regeneration. When parrotfish flourish, the reef attains a dynamic equilibrium balanced between calcification and erosion. When they vanish, entire ecosystems teeter toward collapse, and the sands of distant islands erode into nothingness. In their daily grind, parrotfish embody the paradox of nature's architects, powerful agents of transformation, whose legacy lies not in monuments of stone, but in the shifting sands of life's most intricate habitats. Beneath the low arches of broken coral plateaus, where sunlight filters through scattered rubble in fractured beams, a miniature spectacle unfolds in vibrant technicolor. Here, among the crumbled remnants of dead branching corals and scattered boulders of algal overgrowth, the mandarinfish, Synchiropus splendidus, stages its nocturnal pageant. As daylight retreats, the fish's riotous palette of electric blues, incendiary oranges, and tiger-stripe patterns blooms to life, each scale refracting light through microscopic layered structures that shimmer like prismatic lenses. Rather than emitting their own light, mandarinfish rely on structural color. Stacks of guanine crystals within specialized cells create thin film interference, bending ambient wavelengths into hues so intense they seem otherworldly. This visual alchemy turns the mandarin's body into a living mosaic, a neon war paint designed for intraspecific communication and courtly display rather than mere camouflage. As dusk deepens, two mandarins emerge from adjacent crevices, their fins unfurling in a synchronized wave that billows like silk banners. This courtship choreography follows a strict temporal rhythm. Within minutes of civil twilight, pairs ascend in tandem from the rubble zone, rising vertically toward the open water where currents are weaker. The male extends his elongated dorsal fin into a lance of brilliant blue, while the female mirrors the gesture with a subtler sweep of orange-rimmed pectorals. In these spiraling ascents, the fish maintain a precise spatial gap of mere centimeters, their bodies rotating slowly to exhibit every facet of their color pattern to an audience of potential rivals. Researchers using infrared imaging have documented that these ritual dances can last up to 15 minutes, an endurance feat for creatures barely the size of a person's palm. During the courtship, the mandarin fish's eyes, large and watchful, track one another's movements with uncanny precision, conveying readiness and selectiveness in equal measure. Surprising even seasoned observers, mandarinfish engage in a nuanced role reversal at the moment of gamete release. Unlike many reef species that scatter eggs with minimal parental investment, mandarinfish exhibit a brief pairing phase where both partners circle at the apex of their ascent, vibrating their tail muscles in a final flourish. This tremor loosens a gelatinous matrix of eggs and sperm into the water column, where buoyancy traits carried over from their larval stage ensure a gentle, slow drift. Within hours, the fertilized eggs develop into transparent larvae, each equipped with bioluminescent bacteria that emit faint greenish glows visible under ultraviolet observation. While structural color dazzles the adult mandarinfish, these bacterial luminescence capabilities in larvae hint at an evolutionary past where true fluorescence may have played a larger role in nocturnal signaling and predator confusion. Although adult mandarins lack light-producing organs, the residual glow of their juvenile stage suggests a fascinating ontogenetic shift from biochemical luminescence to purely physical color production. The mandarinfish's flamboyant coloration might invite predation, yet these fish navigate the risk through both behavioral and chemical strategies. Rub surface trials conducted in controlled mesocosms reveal that mandarinfish secrete a thin, mucus coating imbued with subtle toxins, derivatives of terpenoid compounds, that deter many small crustacean predators and cryptic ambush hunters such as small groupers and hawkfish. When a predator approaches too closely, the mandarin remains motionless, its vivid stripes morphing into an aposematic warning rather than a bridge for mimicry. Predators that taste or probe the mucus are met with a bitter, slightly numbing sensation that evokes immediate rejection. Occasional observations of bite and release events by small predatory fish underscore the effectiveness of this chemical defense, suggesting that mandarinfish have negotiated a delicate compromise. They can afford to advertise with color because their skin yields an unappetizing sludge when breached. Even the choice of habitat, rubble zones between living reef and sandy lagoon, reflects the mandarinfish's ecological specialization. These creviced substrates, formed by storms and coral die-offs, provide both hiding places and foraging grounds. With their slender bodies and flattened heads, mandarinfish slip into narrow fissures by day, avoiding the prying eyes of nocturnal snapper patrols. By night, they emerge to graze on the dense biofilm of copepods, amphipods, and protozoans that colonize rubble surfaces. Their small, brush-like teeth efficiently scrape microscopic prey from coral chips and rock faces, turning seemingly barren rubble into a banquet table. In contrast to the open coral crest where larger fish graze on macroalgae, the rubble fields teem with microfauna, offering a rich nutritional niche that mandarinfish exploit with specialist precision. Their foraging also contributes to material turnover. By sweeping away biofilm and dislodging detritus, mandarinfish help maintain the microhabitats that support sponge recruits and juvenile crustaceans, subtly engineering the rubble ecosystem much like larger parrotfish sculpt living reef. Yet these fragmented habitats face mounting pressure from human impacts. Coastal development and sedimentation bury the rubble zones, while destructive fishing methods crush the very structures mandarinfish depend upon. Surveys in the South China Sea reveal local extirpations of mandarinfish in areas where live rock harvesting has cleared rubble piles for the aquarium trade. Such losses represent not only the disappearance of a visual marvel, but also the breakdown of a nocturnal food web in which mandarinfish play a keystone role. Without their nightly grazing, biofilm can accumulate unchecked, altering microbial community composition and increasing disease risk for invertebrates and juvenile fishes, that rely on rubble cover. In this way, the mandarinfish's vivid survival strategies are inseparable from the integrity of their specialized habitat. Predator-prey dynamics in this rubble realm unfold with unpredictable twists. At times, mandarinfish fall prey to mimic species such as the chalk-based butterflyfish, which adopt a similar color palette to intrude on cleaning stations. By resembling mandarinfish patterns, these mimics gain access to parasite-laden hosts or to the rubble crevices where snail prey hides. In response, mandarinfish have refined their recognition acuity, using subtle differences in stripe curvature and scale iridescence to differentiate conspecifics from imposters. Experimental trials employing high-resolution imagery show that mandarinfish ignore decoys with minute pattern deviations, whereas butterflyfish mimic models elicit immediate avoidance behavior. This ongoing visual arms race underscores the evolutionary stakes. While masquerading yields nutritional rewards for mimics, the survival of mandarinfish hinges on the accuracy of intraspecific pattern cues. Amid these ecological dramas, mandarinfish larvae undertake their own remarkable dispersal journeys. Drifting in the upper layers of the ocean for up to 3 weeks, they ride thermocline currents that can traverse kilometers each day. During this pelagic phase, their bioluminescent bacteria provide both a lure and a guard. Faint glows attract cooperative zooplankton that brush against the larvae, assisting them in feeding, while simultaneously deterring voracious jellies and ctenophores that find the luminescence disorienting. As metamorphosis approaches, the larvae shift from vertical migrations, rising at night to feed, to a more horizontal search for suitable rubble fields. Chemical cues released by benthic invertebrates and newly settled corals guide them downward, where they settle and begin the transformation into the neon-brilliant forms that await the reef floor. The transition from translucent, glowing wanderers to grounded, structurally colored adults exemplifies the mandarinfish's two-stage strategy. Roam, evade and feed in the open ocean, then dazzle, seduce, and maintain the rubble realm. By the time moonlight spills across the rubble zone, the multiplied colors of dozens of mandarinfish create an almost hypnotic pattern, a shifting tapestry that ebbs and flows with the rhythm of nocturnal life. Shrimp and cryptic crabs scuttle amid the moving brilliance, emboldened by the mandarinfish's chemical cloak, which masks their own, rendering them less detectable to hunting eels and rays. Observing through low-light cameras, researchers note that predator strike frequency in mandarinfish territories is half that of adjacent sandflats, a testament to the combined power of visual intimidation and toxic deterrence. Even so, mandarinfish vigilance never ceases. They patrol their microdomains in small, rotating groups, ensuring that no predator patterns a successful ambush location. In these fractured reefs, destitute of grand coral edifices, yet rich in rubble complexity, mandarinfish embody the adage that beauty can be survival's sharpest tool. Through structural color that manipulates light, ritualized dances that bond partners and signal rivals, chemical defenses that turn flesh into sludge, and an uncanny specialization for life among the reef's debris, these fish have carved out a niche both spectacular and precarious. Their survival underscores the importance of microhabitats often overlooked by conservationists, reminding us that the most dazzling performances often unfold on the smallest stages. In the neon glow of the rubble zone, the mandarinfish's psychedelic show continues night after night, a living testament to nature's ingenuity and the fragile balance that sustains it. Beneath the wavering surface of turquoise shallows, a series of radial patterns in the sand betray an unseen drama. As currents shift and cast subtle shadows across a once thriving patch reef, an unwelcome guest slips through the fractured crests of Montastria colonies, the lionfish. Its body, adorned with alternating bands of crimson and ivory, moves with deliberate stealth, fins outstretched like a phantom's cloak. In a single fluid motion, the fish kneels on its powerful pectorals, angling its head toward the reef floor and tallying the unsuspecting shrimp and juvenile fish that dart among rubble and coralline algae. Then, as if triggered by an invisible signal, it pounces, lunging forward in a blur of barbed spines that close an invisible trap around its prey. This ballet of ambush, honed over millennia on its native Indo-Pacific grounds, plays out here on Caribbean reefs with devastating effect. An ecological dagger thrust into ecosystems unprepared for its assault. The lionfish's venomous anatomy represents a masterclass in biological weaponry. Each dorsal spine bears a dual-channel groove that houses venom-producing glands, while the opercular and pelvic spines add lateral defense. When threatened, or when subduing prey, the lionfish depresses these spines against elastic sheaths, exposing serrated needles that inject a complex cocktail of proteins and neurotoxins. Among these, neurotoxins interfere with ion channels in nerve membranes, causing immediate pain, paralysis, and in larger doses, muscular convulsions. Observers who have suffered accidental stings describe searing heat radiating from the puncture site, followed by swelling that can obscure the spine's entry points. Researchers analyzing venom samples have identified additional myotoxins that break down muscle tissue, ensuring that any predator foolish enough to swallow a spine-laden lionfish will endure internal tissue damage. In the absence of natural predators here, these spines serve as both spear and shield, enabling lionfish to roam Caribbean reefs with impunity. Even more insidious than their defenses is the lionfish's predatory strategy. Unlike many reef hunters that drive prey into confined spaces, lionfish employ flare and funnel tactics. They spread their enormous pectoral fins to corral small fish and invertebrates against reef walls, cutting off escape routes. As they advance, they puff their heads and erect dorsal spines to appear larger, exploiting the instinctive caution of their quarry. Once the prey is sufficiently cornered, a swift vacuum of bony jaws inhales the victim whole. Field studies employing underwater video have captured lionfish consuming prey up to half their own body length, with a single individual capable of devouring dozens of prey items in rapid succession. These feeding bouts disproportionately target juvenile parrotfish, damselfish, and wrasses, species essential for algal control and coral recruitment, thereby weakening the reef's foundational processes. The ecological havoc wrought by lionfish extends far beyond direct predation. Within months of their introduction in the mid-1980s, lionfish populations exploded along Florida's Atlantic coast and throughout the Caribbean, unchecked by parasites, predators, or competitors. The resulting trophic cascade has unfolded like a slow-motion collapse. As grazing fish decline, macroalgae proliferate, shading out corals and altering reef structure. Juvenile corals deprived of the open substrate cleared by parrotfish and surgeonfish struggle to establish, leading to reduced reef complexity and diminished fish habitat. In turn, commercially important species such as groupers and snappers, whose adults rely on healthy juvenile nurseries, begin to falter. Long-term monitoring sites in the Bahamas have recorded lionfish densities exceeding 400 individuals per hectare, orders of magnitude higher than native predator hotspots, correlated with 65% reductions in small reef fish biomass. This chain reaction underscores how a single invasive predator can rewire an entire ecosystem, shifting it toward algal dominance and impoverished biodiversity. Faced with this relentless invader, Caribbean communities and conservation organizations have mobilized control crusades that blend science, local stewardship, and even culinary innovation. Spearfishing tournaments, lionfish derbies, call on divers to hunt as many lionfish as possible, with prizes awarded for size and count. The harvested fish are then funneled into restaurants and markets, reframing lionfish from ecological scourge to culinary delicacy. Chefs tout the firm white flesh as mild and flaky, encouraging consumers to eat them to beat them. At research stations, scientists have refined capture tools, such as specialized nets and modified traps, that limit bycatch and minimize habitat damage. In parallel, experimental studies of enzyme-based deterrents seek to exploit lionfish chemical communication channels. By introducing synthesized alarm cues from wounded conspecifics into lionfish habitats, researchers aim to disrupt feeding aggregations and reduce local densities. Early trials show promise, with alarm treatments causing transient schooling and flight responses, though scaling these methods across vast reef systems remains a challenge. Meanwhile, marine protected areas have begun integrating lionfish control into broader reef management plans. Sites such as the Bonaire National Marine Park deploy dedicated lionfish response teams whose members survey transects daily, removing individuals before they can establish breeding populations. Tagging efforts reveal that lionfish exhibit site fidelity, often returning to the same crevice after removal, which allows for targeted, efficient culling. Genetic analyses of Caribbean lionfish populations, however, trace their spread back to a small number of founder individuals released in Florida, a stark reminder of how singular human actions can precipitate scale-shattering biological invasions. This genetic bottleneck also offers a glimmer of hope. With limited diversity, lionfish may be more vulnerable to emergent diseases or targeted biological controls, though the ethical and ecological ramifications of such interventions demand careful consideration. In the heart of these control efforts lies a lesson in resilience and collaboration. Divers in Barbados share real-time lionfish sighting data through mobile apps linked to researchers, enabling rapid response. Local schools incorporate lionfish biology into curricula, fostering the next generation of reef stewards. Remote villages in Belize that once relied on fish traps to harvest spiny lobsters now adapt their techniques to capture lionfish, gaining both food security and ecological benefits. At the same time, international networks of scientists convene annual workshops to refine best practices, share detection methods using environmental DNA, and model lionfish population trajectories under varying removal regimes. As sunlight wanes and the reef settles into twilight, the lionfish that remain blend into the shadows, their vertical stripes disrupting the contours of crevices where they lie in wait. Yet for every lionfish removed, another juvenile drifts in on the currents, testament to both the species' reproductive potency and the persistence required of human defenders. In this contest, victories are measured not in eradication, that razor edge remains elusive, but in balance, reducing lionfish densities to levels where native assemblages can recover, where algae retreat, and where coral recruits find space to grow. The lionfish invasion, stark in its simplicity and sweeping in its impact, underscores the fragility of marine networks and the far-reaching consequences of human introductions. By confronting this technicolor predator with collective ingenuity, communities across the Caribbean demonstrate that ecological redemption, even in the face of a perfect invader, is possible when science, policy, and local action converge beneath the waves. Beneath the muted glow of early morning light, the seabed emerges from darkness in muted tones of olive and slate, punctuated by the occasional flash of porcelain coral and swaying strands of seagrass. In this subdued realm, a solitary pufferfish patrols the sandy plain, its mottled skin blending seamlessly with the substrate, until a curious ripple reveals its presence. As it cruises forward, black eyes scanning for morsels, the pufferfish's body appears deceptively soft and rounded, until it senses a passing current disturbance, the shadow of a hunting grouper sweeping overhead. In an instant, the fish's form balloons outward in a perfect sphere, its normally lax skin stretching taut over a swollen internal cavity. The transformation happens within seconds. A rapid intake of water, sometimes air if near the surface, forces its stomach walls to expand, while its spines, previously flattened against its flanks, stand erect in a defensive display. This extraordinary inflation, a mechanical feat of skeletal flexibility and hydrostatic pressure control, erects an almost impenetrable barrier. Any predator that attempts to swallow the inflated pufferfish is met with a prickling wall of spines and an unyielding bulk that lodges in the throat. Watching this in ultra-high-speed footage, one notes that the pufferfish's vertebral column is uniquely adapted with interlocking yet flexible joints that permit expansion without compromising structural integrity. Muscles along its abdomen contract and relax in precisely coordinated sequences to manage the inflow and retention of water, while specialized valves in its digestive tract prevent backflow. Researchers measuring internal pressures have recorded spikes up to 5 times greater than ambient water pressure, a testament to the fish's remarkable physiological control. Yet inflation is only half of the pufferfish's defense. It is the toxin coursing through its tissues, tetrodotoxin, that turns this fish into a walking biochemical landmine. Unlike many marine toxins produced for immediate stinging or envenomation, tetrodotoxin operates at the cellular level, binding irreversibly to voltage-gated sodium channels in nerve and muscle cells. In blocked channels, nerve impulses cannot travel, causing paralysis that spreads rapidly throughout an attacker's nervous system. For a predator that miscalculates its risk, the result is swift immobilization, often followed by respiratory collapse. The origins of this potent toxin have long been debated among scientists. Initially thought to be synthesized endogenously by pufferfish themselves, recent molecular studies suggest a more intricate partnership. Bacterial symbionts residing in the fish's gut and skin glands produce the bulk of tetrodotoxin, which the pufferfish then bioaccumulates and concentrates within specific tissues. Strains of Vibrio and Pseudomonas bacteria, identified through genomic sequencing, harbor the enzymatic pathways required for tetrodotoxin synthesis. Experimental pufferfish, reared in sterile aquaria, devoid of these bacteria, remain non-toxic. a striking demonstration that without their microbial allies, puffers lose their chemical shield. Yet the question of how these bacteria arrive in wild pufferfish remains open. Larvae drift through ocean currents until they encounter environmental reservoirs of toxin-producing microbes, embedding them within their developing tissues. This symbiotic acquisition underscores a hidden layer of ecological interdependence, where a fish's survival relies upon microscopic partners it never directly cultivates. As the morning current settles and the inflated pufferfish deflates itself back to its streamlined shape, it resumes grazing. With a beak-like arrangement of fused teeth, the fish chips away at the calcareous tubes of marine worms, snaps at tiny crustaceans, and nips at mollusk shells, crushing prey in swift, precise bites. These feeding actions demonstrate the pufferfish's dual nature, part artful engineer of defense, part opportunistic forager. Yet for humans, the pufferfish's most notorious role emerges in coastal Japan, where it is transformed into fugu, a culinary gamble measured in molecular micrograms. In specialized restaurants, chefs undergo rigorous apprenticeships, often exceeding 2 years, learning to excise the lethal organs where tetrodotoxin concentrates, the liver, ovaries, and skin glands. Even the precise location and minute depth of incisions matter enormously. A few milligrams of toxin can kill an adult human. Regulatory bodies mandate licensing examinations that include both written tests on toxin biochemistry and practical assessments involving fish dissection. Despite these safeguards, the occasional miscalculation and inadvertent nick into a toxin-laden membrane can have fatal consequences. Yet paradoxically, fugu remains a celebrated delicacy, prized both for its delicate texture and for the thrill inherent in its potential danger. The cultural fascination with pufferfish toxin extends beyond gastronomy. Folk traditions have long regarded tetrodotoxin as an aphrodisiac or spiritual elixir. Although modern scientific analysis debunks such claims, the substance's effects are purely paralytic with no psychotropic properties. However, preliminary medical research explores tetrodotoxin's analgesic potential, harnessing its sodium channel blocking for pain relief in conditions like cancer neuropathy. Clinical trials have administered purified toxin under controlled dosage, achieving significant pain reduction without permanent paralysis. These investigations illustrate how evolutionary weapons can, under strict human oversight, transform into therapeutic tools. Another testament to the pufferfish's unexpected contributions beyond its aquatic realm. In the wild, the pufferfish's toxin and inflation mechanics have driven predators into adaptive arms races. The tiger grouper, one of its few potential threats, has developed specialized hunting strategies, ambushing pufferfish when they are gravid or during spawning aggregations, times when the fish's bodies cannot inflate fully without jeopardizing their reproductive tissues. Video recordings from remote reef cameras show grouper stalking pregnant female puffers in shallow sandy gutters, striking with precision before the fish can execute their full defensive routine. Similarly, certain species of shrimp have evolved tolerance to sublethal doses of tetrodotoxin, scavenging on pufferfish carcasses and thus playing a role in toxin redistribution across the benthic community. These scavengers often bear traces of toxin in their own tissues, demonstrating how tetrodotoxin lingers in the ecosystem, challenging the principle that toxins are neatly compartmentalized. The evolutionary interplay deepens in pufferfish mating displays. Males of some species sculpt intricate geometric circles in fine sediment, sometimes over 2 meters in diameter, using rhythmic fin strokes to carve ridges and grooves that catch planktonic particles. These crop circles not only function as nest sites for spawning, but also serve as visual advertisements of male fitness. Females inspect these constructions, swimming through the central troughs and assessing the precision of the patterns before releasing eggs, which the male immediately fertilizes. The complexity of the design correlates with the male's body size and stamina, traits linked to toxin load and offspring viability. In this way, tetrodotoxin mastery and reproductive success intertwine. Males with higher toxin concentrations likely indicate robust, symbiotic bacterial communities and superior immune competence, qualities females may seek to pass on to their progeny. As afternoon shadows lengthen across the reef, the pufferfish drifts into a clearing of seagrass, its mottled skin becoming nearly invisible against the waving fronds. A juvenile reef shark glides nearby, drawn by curiosity and the faint scent of potential prey. The pufferfish remains still, strategic immobility augmenting its other defenses. Should the shark attempt an attack, the fish will inflate, presenting spikes and a chemically dangerous surface. Yet the shark often retreats, its evolutionary memory ingrained with caution toward these potent fish. This behavioral stillness, relying on crypsis until the moment of maximum defense, complements inflation as a two-tiered survival tactic, conserving the fish's energy while minimizing unnecessary confrontations. By dusk, the pufferfish seeks shelter beneath an overhanging ledge, its fins flexing gently to maintain position. Microhabitat selection plays a crucial role. Crevices must be large enough to accommodate inflation without risking entrapment, yet snug enough to deter larger predators. At night, however, puffers face additional threats from venom-resistant eels and octopuses that can probe crevices with probing arms or slender jaws. Some pufferfish have been observed secreting extra quantities of mucus as an additional chemical barrier, hinting at real-time adjustments in toxin deployment based on perceived risk. These nuanced behaviors highlight the pufferfish's capacity for situational defense modulation, a cognitive dimension often underestimated in fish. Across the reef gradient, from shallow flats to deeper slopes, pufferfish species exhibit dietary and morphological specializations that reflect variations in toxin allocation and inflation reliance. Deepwater species possess fewer and shorter spines, but tend to have higher toxin concentrations, trading mechanical defense for chemical potency in darker, less structured habitats. Contrastingly, reef edge species favor robust spines and moderate toxin levels, balancing the need for inflation in open areas with the metabolic costs of toxin maintenance. This geographic mosaic of defense strategies exemplifies how evolutionary pressures shape the pufferfish's dual arsenal in response to habitat complexity and predator communities. As moonlight filters through the water's surface, the pufferfish settles into its nighttime refuge, neither fully inflated nor completely deflated, maintaining a slight engorgement that primes its defenses. Sand grains shift around its body, settling into a partial cloak of camouflage. In this transitional posture, the fish consolidates its toxin stores, renewing bacterial symbiont populations through secretions in its skin and gut. This nocturnal regeneration ensures that by dawn's first light, the pufferfish remains both physically and chemically prepared for the challenges ahead, ready to expand its form, unleash its potent neurotoxin, and persist as the ocean's ultimate biochemical threat. In the silent interplay of predator and prey, the pufferfish stands unrivaled, a living testament to the power of symbiosis, physiological innovation, and the evolutionary arms race that shapes life beneath the waves. Beneath the green-dappled surface of a tidal inlet, a field of eelgrass sways like an emerald forest in a gentle breeze. Here, amid the narrow blades of Zostera marina, a single seahorse hovers with deliberate stillness, its prehensile tail coiled tightly around a strand of grass. The curvature of its spine aligns perfectly with the blade's arc, minimizing drag as the current ebbs and flows. Each pulse of water through this nursery habitat sends ripples of sediment that briefly cloud the seagrass roots, yet the seahorse remains unwavering, anchored by a tail whose musculature and flexibility rival the finest grappling hook. Observing its hooked grip, one notes the subtle adjustments. The tail's segmented rings flex and slide to redistribute pressure, preventing fatigue during hours-long station keeping. In these moments, the seahorse transcends its small stature, assuming the role of a vigilant sentinel, eyes scanning in all directions, head pivoting on its bony joint like a turret, ever watchful for drifting plankton and lurking predators alike. As morning light filters through sunlit ripples above, the seahorse releases its hold with a measured uncoil and drifts upward, fins beating in rapid quivers to counter the vertical current. Its head remains motionless relative to its body, a stabilization strategy honed by its labyrinthine inner ear and visual processing centers. In this way, the creature scans for copepods and tiny crustaceans that drift past in the water column. When prey enters reach, the seahorse executes its signature snap feeding tactic. Without warning, it contracts trunk muscles to create a vacuum within its tubular snout, sucking in food items at speeds rivaling the lash of a mantis shrimp's claw. Because of the seahorse's lack of true teeth or pharyngeal mill, prey must be small enough to pass through its narrow mouth. Yet this feeding method proves astonishingly efficient. Research cameras have captured successful suction strikes in under 6 milliseconds, a testament to evolutionary precision. Later in the season, this same sentinel transforms its solitary vigilance into a performance of devotion. When a gravid male seahorse arrives at a preferred patch of seagrass, he displays subtle color shifts, pale gray spots morphing into deeper russet hues, signaling readiness to receive eggs. The female approaches in a mirror of caution, aligning her tail alongside his and depositing a ribbon of several dozen eggs into the brood pouch lining his abdomen. Here, within a sealed, vascularized chamber, the male undertakes a physiological feat unequaled among vertebrates. He provides oxygen and nutrients to developing embryos while regulating osmotic balance in a fluctuating saline environment. Capillary networks in the pouch wall invite gas exchange, drawing oxygen from his bloodstream and offloading carbon dioxide into the surrounding water. Concurrently, he secretes specialized ions and proteins that maintain the egg's internal milieu, ensuring embryos neither desiccate nor overinflate, as external salinity oscillates with the tide. Over the following weeks, the male's transformation deepens. His skin near the pouch becomes opaque, dotted with fine ridges that indicate expanding embryo clusters. Under low-light endoscopic cameras, researchers have observed that males can modulate brood size post-deposition, reabsorbing or expelling underdeveloped eggs to match their own energetic reserves and environmental conditions. This selective brood control underscores a critical evolutionary adaptation. In periods of scarcity, reducing clutch size improves survival odds for remaining offspring rather than risking all on an unpredictable habitat. At night, while most seahorses rest in the shelter of seagrass tangles, the brooding male remains partially active, instinctively clutching a sturdy holdfast to stabilize himself against nocturnal currents, his respiration rate adjusting to maintain optimal oxygen levels for both himself and his young. Yet the seahorse's ecological niche, so finely tuned to these seagrass habitats, faces relentless pressures. Coastal development clears shallow shoals for marinas and real estate, uprooting eelgrass meadows that once stretched uninterrupted for miles. Where seagrass beds have receded, sedimentation smothers remaining blades, depriving seahorses of both camouflage and anchoring sites. At the same time, poaching for traditional medicine, curios, and the aquarium trade further decimates populations already constrained by low fecundity. Most species produce only a few hundred young per breeding cycle, and juvenile mortality often exceeds 90% in the wild. Genetic surveys on the endangered spotted seahorse reveal alarmingly low diversity in fragmented populations, suggesting that inbreeding and local extirpations are reducing resilience to disease and environmental change. Each uprooted seagrass shoot, each illegal collection, chips away at the seahorse's ability to sustain its next generation. In response, citizen scientists and conservationists have forged a multi-pronged defense. Aquaculture facilities now breed seahorses under tightly controlled conditions, replicating tidal salinity fluctuations and providing natural prey to ensure robust broodstock health. Through carefully monitored releases, captive-bred juveniles bolster depleted wild populations, with site selection guided by habitat suitability models that factor in water clarity, seagrass density, and predator abundance. Additionally, global tagging programs employ miniature acoustic transmitters, encased in biocompatible resin, that attach gently to the seahorse's dorsal ridge. As tagged individuals move through seagrass corridors, arrays of underwater microphones record their exact positions, revealing migration pathways, habitat preferences, and seasonal congregation sites. These data inform protected area boundaries and guide restoration efforts, ensuring that newly planted eelgrass patches align with the seahorse's natural range. One remarkable outcome of these collaborative efforts emerged in a bay where eelgrass restoration and seahorse reintroductions coincided. Within 2 years, juvenile seahorse sightings increased tenfold, while local fishers reported more vigorous populations of small crustaceans and juvenile fish, species that depend on the structural complexity seahorses help maintain through their feeding. In parallel, local schools integrate seahorse monitoring into their science curricula, equipping students with trays of marked juveniles and digital cameras to log developmental milestones and habitat use. This grassroots engagement not only supplies long-term observational datasets, but fosters stewardship among the next generation, who come to view seahorses not as distant curiosities, but as integral components of their coastal heritage. Across deeper channels and tidal creeks, researchers continue to uncover fresh insights into seahorse biology. Recent tagging studies revealed that some species undertake nightly foraging migrations of up to half a kilometer, moving from dense seagrass meadows to adjacent mangrove roots where hydroid colonies thrive. These unexpected excursions underscore the seahorse's role in linking disparate habitats, transferring nutrients and influencing microfaunal communities across ecosystem boundaries. It also highlights the complexity of designing marine reserves. Protecting only seagrass beds may prove insufficient if seahorses regularly exploit off-reef structures during crucial life stages. Returning focus to the solitary sentinel, one observes a fully brooding male gently rocking atop a seagrass stem as dusk falls. Its tail maintains a firm coil while its eyes, independently swiveling, scan the dimming water for signs of nocturnal predators. Around him, plankton pulses toward the surface, and distant silhouette shadows of rays gliding overhead stir faint eddies. Within his pouch, dozens of embryos paddle against the chamber walls, preparing for an imminent release timed with the lunar cycle. When the moment arrives, the father will expend remarkable energy to expel each fragile juvenile into the open water, a final act of paternal sacrifice that entrusts the next generation to the whims of currents and invisible microbes. In the unhurried twilight of this seagrass forest, the seahorse's life cycle, from prehensile anchoring to paternal incubation, reveals an intricate tapestry of adaptation and vulnerability. Its survival hinges on the persistence of submerged meadows, the vigilance of guardians both piscine and human, and the continued collaboration of scientists, local communities, and citizen observers. As restoration efforts expand and seahorse tracks illuminate hidden corridors beneath the waves, there is reason to hope that these horses of the sea will continue their silent patrols, steadfast sentinels of the shallow coastal realm they call home. Beneath a sky of fractured light, where sunbeams dance across the rolling contours of a coral metropolis, the reef reveals itself as a vast battlefield of survival strategies. Here, among the undulating gardens of staghorn coral and the sculpted towers of brain coral, 9 champions stake their claims, each armed with a unique suite of weapons, defenses, and alliances that together form the living arsenal of the reef. As the current carries suspended particles through this underwater panorama, the stage is set for an epic survey of nature's ingenuity, from the razor-sharp spines of the blue tang to the psychedelic color blitz of the mandarinfish, from the armored cocoon of the parrotfish's nightly refuge to the fatherly tenacity of the seahorse brooding against shifting tides. Patrols of spiky sentinels intersect with schools of agile grazers. In the midday glare, the clownfish weaves through anemone tentacles, its mucus shield neutralizing stings in a chemical détente. Nearby, the blue tang slashes at turf algae with hidden razor spines, maintaining the reef's hygiene with surgical precision. The angelfish follows its own turf management protocol, balancing the removal of harmful algae with selective grazing on coral polyps, a delicate calculus that sustains both its diet and the substrate for new coral recruits. Each species wields its feeding apparatus as both weapon and tool, the parrotfish's beak pulverizing dead coral into sediment that molds the reef's topography, the butterflyfish's patterned hide-fracturing predator's focus, the pufferfish's inflation and tetrodotoxin, combining a mechanical barricade with a biochemical minefield. Together, they punctuate the reef's profile with a kaleidoscope of survival designs, each honed by evolutionary pressures that demand both offense and defense. Yet none of these strategies exists in isolation. As noon turns to afternoon, the reef's social networks become more apparent. Cleaner shrimps beckon the butterflyfish for parasite removal, their porcelain claws plucking irritants with surgical care, while corkscrew anemone arms shelter the clownfish, sustaining a partnership that elevates both participants. Parrotfish grazing opens substrate for sponges and juvenile corals to settle, creating new real estate that in turn hosts wrasses and gobies, which reduce microalgal overgrowth and strengthen water circulation around coral heads. Where mandarinfish vibrantly display for mates, they also stir biofilm, feeding small invertebrates that become prey for larger grazers and predators, closing loops of energy transfer. Even the invasive lionfish, though an ecological scourge, has inadvertently spawned human-led alliances. Dive teams and coastal communities now converge in lionfish derbies, spears in hand, forging new social bonds across international lines to combat a common threat. These interwoven relationships constitute a reef alliance, a dynamic web built on reciprocity and exchange, where each species' actions resound through the community. As the sun dips toward the horizon, the reef's resilience is tested by the mounting pressures of a warming ocean. Thermal anomalies trigger mass bleaching events, stripping corals of their symbiotic algae and erasing the very scaffold that supports this intricate fortress of life. Where temperatures cross critical thresholds, anemones expel banded fish, butterflyfish lose the contrast that masks them, and parrotfish cocoons fail under oxidative stress, leaving sleepers exposed to nocturnal predators. Ocean acidification weakens the skeletons of stony corals and the shells of microbial prey, forcing grazers like the mandarin and blue tang to shift diets and grazing patterns, sometimes with detrimental ripple effects on nutrient cycling. Overfishing removes key browsers, allowing macroalgae to overrun cleared spaces, and habitat destruction chops seagrass meadows that seahorses, the masters of stealth and paternal care, depend on for anchorage and nursery grounds. The balance that took eons to refine now tilts precariously, favoring opportunistic species at the expense of mutualists that sustain the reef's complexity. Yet the story need not conclude in decline. Across the globe, scientists, communities, and governments are enacting measures that embody the reef's own lessons in cooperation. Restoration projects employ microfragmentation techniques to propagate resilient coral genotypes, while larval seeding replicates the angelfish's long-distance dispersal, ensuring genetic connectivity across broken reef networks. In the Caribbean and Indo-Pacific alike, community dive patrols monitor lionfish populations and transplant nursery-raised parrotfish to bolster grazing pressure. Innovative marine protected areas now include dynamic zoning that reflects the nightly migrations of seahorses and the pelagic drifts of mandarinfish larvae, safeguarding habitats critical at different life stages. Satellite-linked acoustic receivers and citizen science apps map the movements of tagged blue tangs and butterflyfish, feeding real-time data into adaptive management systems. Even the illicit aquarium trade is giving way to sustainable aquaculture. Captive breeding of clownfish, blue tangs, and mandarinfish reduces wild capture, while toxin harvesting research into pufferfish's bacterial symbionts offers medical breakthroughs without endangering wild populations. In this octave of oceans, the 9 species emerge not merely as isolated case studies, but as soloists in a symphony of survival. Their weapons, spines, toxins, camouflage, bioerosion, interlock to produce a score of ecological functions that maintain the reef's harmony. Their alliances, mutualisms, territory maintenance, cleaning stations, compose the movements that regulate energy flow and community structure. And their collective response to threats illustrates a crescendo of ingenuity. When one instrument falters, others adapt or amplify, preserving the overarching composition. The reef's future will depend on our ability to extend this paradigm of cooperation beyond its underwater realms, integrating social, scientific, and policy-driven movements that mirror the reef's own strategies. By embracing the multifaceted roles of reef champions, from clownfish diplomats to seahorse sentinels, we recognize that protecting marine ecosystems is not a solitary pursuit, but a symphony requiring diverse voices, concerted actions, and a shared commitment to sustain life's most vibrant stage beneath the waves.